Hi there, my name is Josh Proehl and I'm a ninth grader at Newland and Spicer High Schools, and I'm doing a research project on the effects of chemical Irgasan and Amphipods. The idea of this project is to see what the effects of Irgasan, which is a man-made chemical that was made in the laboratory, has on aquatic amphipods, which are insects that are vital to the ecosystem in lakes and rivers. This chemical has been showing up on water tests recently that have been done, and it doesn't dissolve in water, which is why it's involved in antibacterial soaps. And when you wash your hands with it, it goes straight down the drain and just sits in the lakes and rivers, and nobody knows what it does yet, so that's what the purpose of this experiment is, to find out. Like a lot of people, I like to go fishing. So if this chemical were to affect fish, or the fish is the minnow's food source, then eventually along the line it's going to keep affecting the entire lake, and we're going to have problems not only with the animals that live in the lake, but as well as all the animals that drink water from that lake. So it would just kind of collapse an ecosystem that we want. My question was, will the chemical Irgasan, which is a household chemical, affect the behavior rates or the mortality of the scuds or the amphipods that I'm using? And I planned this out, and I predicted that the mortality rates would greatly increase and that the behavior rates would decrease, so they would be moving a lot less and many of them would die throughout the experiment. After I formed my question, I had to look into how I was going to set up this experiment. And so I looked deeper into some standards that were around, and I found the EPA, or the Environmental Protection Agency. And they do a lot of this testing with the scuds and fish and other animals that they need to test their chemicals on. So I did what they would do if they had an aquatic animal. They keep them individual containers with a 2 to 3 day trial, and then that way they're all separate, they're quarantined from each other. Each one will have its own results, and there won't be any cross-contamination. And afterwards, if they are affected or if they're not, they have to freeze them so that they can't just release them, and that's the same process that I do so that they aren't spread in the wild or anything. Now, to test this, I set up 60 containers each with 15 in each group. Each group contains a varying level of chemical, starting from no chemical in it to be our control all the way up to 2,500 nanograms. And nanograms is like a parts per million, so it's a very small amount. And since the chemical Irgasan comes in a powder form, we have to use a diluted solution. And we have that mixed into our water bucket here, and now we have to measure out 200 milliliters for each scud, and they'll be each in individual containers and monitored through a period of 3 days. And then after these 15, there'll be 15 more, and we'll repeat it 4 times so that we have a total of 60 scuds on each level. So I'll just measure out the milligrams of water we need, and then I'll add a scud from the bucket. And so now it's set and ready to go. Once I start the research, the project will take 12 days of actual data collecting experiment, and then throughout the process of analyzing and getting it all put together, finding what we have and typing it all up, putting it onto a board, it'll probably be another month after that before it's finally completed and science fair ready. I think the hardest thing that will be in this project will be going through and seeing each individual scud, because during setup of this project each one seems to have a little bit different personality. Like, there's a general idea for what scuds do, but it's almost like I might have to end up keeping tabs on individual scuds' personality, because they all seem to have a different behavior pattern from the one right next to it. when I finish my project, spreading it is the fun part, the easy part that I really enjoy doing. I really enjoy going out and telling people about what's about my project. I mean, I'd tell it to anybody that would be willing to sit down and listen to me talk on for a while. Spreading the word is a part that you need in the cycle. I mean, what's the point of doing the entire scientific cycle if you're just gonna make a project and let it sit in your basement? You gotta, once it's done, you have to get it out there. You have to let the world know what you've done. Show them your results. Let them know what stuff's happening that they might not even know about.